Hi, my name is Jack Zilligan.
I'm Naren Prasad.
I'm Jack Dietz, and we are the best
Biotech committee.
Today, we will explain how RNAi can silence a gene in C. elegans. The C. elegans are essentially little worms that eat bacteria. We will be giving them certain bacterial strains to change their phenotype and silence a gene within them. The purpose of this lab is to study the C. elegans and observe the differences between the phenotypes in the wild type and the altered C. elegans. At the beginning of the experiment, we had regular C. elegans feeding off the OP50 E. coli strands, which was the wild type worms or the control group.
We had to get the C. elegans into a separate petri dish without contaminating them. First, we bleached the worms with a drop of bleach to kill the adult worms. Then, we added a drop of the M9 solution to pipette the worms out of the original petri plate.
We swirled the M9 buffer around to attach to the worms and then pipetted 1,000 microliters of the solution with the worms to the other petri plates containing the certain bacteria strains with double-stranded RNA. These new plates contained ampicillin to kill off all of the bad bacteria and IPTG, which makes the bacteria reproduce. Once in the other petri plate, we bleached the worms again with 1 drop of bleach to kill the remaining adults and sealed the petri plates with parafilm to contain the life inside. This is the plate of worms we added the dumpy double-stranded RNAi to. As you can see, the dumpy RNA causes the worms to be short and fat. This change in the phenotype for the dumpy starts to show up around the adult stage of the worm. We think that the dumpy strand turns off the growth genes in the C. elegans, causing the worm to stay short and stubby.
Here is the plate of worms affected by the UNC bacteria. The UNC bacteria causes the worms to start violently shaking, and these phenotypes begin to show up around stage L4 and when they're adults. Seizures are usually caused by excessive electrical activity in the brain. If the UNC bacteria causes the worms to have a seizure, then the gene it is affecting must cause the worm to have electrical stability in the brain and keep the worm safe from seizures and violently shaking.
Finally, here is the plate of worms affected by the blister strand of RNA. The worms that were affected by this strand had a phenotype of a huge blister on them. In humans, blisters are caused by damage to the epidermis through heat and friction. Therefore, the blister strand of RNA must turn off the gene to repair its outer layer.
Initially, the double-stranded RNA goes into the cells and triggers the RNAi pathway, which allows for the process of silencing the gene to occur.
Then, the double-stranded RNA and the interfering RNA is chopped up by the dicer.
The small interfering RNA form a RISC complex, RNA-induced silencing complex, by binding to proteins.
After that, the double-stranded RNA is unzipped, which leaves them single-stranded and allows for the RISC to probe the cell for a recognizable mRNA.
The RISC binds to this mRNA in order to cleave it and makes a mirror image of the mRNA.
At this point, the mRNA can no longer be translated into a protein, and thus the gene is silenced.
Not all of the RNA is chopped, just a significant amount. We learned so much from this lab, including learning how to work with model organisms
And how to work with sterile procedures.
Thank you so much for watching.